Alright, so in this video, I'm going to take you through the inflammatory and immune response using this general here. We're going to talk about how the body responds to cellular damage and foreign antigens. So we'll get back to this and talk about more of these guys individually and how they relate to the overall model. But let's talk about the players involved, which are the white blood cells. The official term for white blood cells are the leukocytes, and white blood cells don't fall into just one thing, like just red blood cells. But white blood cells, there's many different types, such as these mast cells, which release chemicals to start inflammation. Neutrophils, which are the bacteria slayers that ingest and destroy pathogen or invaders. The eosinophils do the same kind of thing as neutrophils, but they particularly are focused on antibody-coated parasites. The monocytes are macrophages. They ingest and destroy invaders, but they're also really important for presenting antigens. We also have these plasma cells or other lymphocytes, the B cells, the T cells. So they're going to have specific responses because they are seeking out and looking at surface antigens. But the plasma cells are also involved with antibody production. We also have some dendritic cells that are located in various spots around your body that are kind of more anchored, and they can also recognize pathogens and activate other cells similar to the antigen presentation with macrophages. So all this stuff comes from all these different types of white blood cells, and ultimately red blood cells as well come from stem cells. And specific types of stem cells called hemo for blood, cyto, cell, blast is to make. So hemocytoblasts, which exist in the flat bones of adults and also into some leg bones of kids. But these, your bone in your bone marrow are these cells that are going to end up making your blood cells, white blood cells and red blood cells. So where in the heck are these white blood cells located? In connective tissue, typically. As you can see in this very generalized connective tissue picture, you've got your fixed macrophages and other white blood cells. You've got free macrophages, you've got mast cells, but then you've also got other cells present, and you also have a bunch of these fibers that are cutting through here in connective tissue, and you have ground substance that the fibers and the ground substance make up the matrix. So when we're talking about connective tissue, we are talking about everything from blood to bone. It's made up of cells, fibers, and ground substance. And connective tissue is classified according to the ground substance, whether it's liquid, syrupy, firm, or gel-like, or if it's solid. Now, again, as we're looking at this, we are bringing up ideas like lymph and lymphoid organs. And we also have this syrupy or loose connective tissue, which is filling in spaces in between organs. So if we're looking at like different coloring books here and lymphatic tissue, you have an entire system of vessels that runs adjacent to all of your blood vessels that take interstitial or extracellular fluid back to and clean it and then drop it back into your blood vessels. So you've got all these lymph nodes. Maybe you've had doctors feel them in your neck to see if they're swollen, but you also have a large conglomeration of lymph nodes in your armpits and your inguinal region. So again, you have all of these things these vessels that get pumped through muscle contraction and move all this large, all the fluid back in here, and then it gets dumped back into your subclavian vein. But if we look at the structure of lymphoid organs, like your lymph nodes, even though we're going to be looking at underneath the skin, you have a large amount of these leukocytes or white blood cells focused into lymph organs, which are a huge part of your immune system. Again, we're going to be focusing on your skin, and we've got our connective tissue underneath this epidermis. So in your dermis here, you've got a lot of loose areas here. In your epidermis, it's cell on cell and it's really packed. If we get through there and underneath here, that's the area we're going to be focusing on with our model. So again, if we flip that around, here's your skin. You've got mast cells, you've got macrophages, you've got helper T cells, killer T cells, B cells, and then in our blood vessels, we also have neutrophils and eosinophils, as well as red blood cells or erythrocytes in your blood vessel. So let's get into the story ones. Once upon a time, there was human tissue maintaining homeostasis, just keeping everything hunky-dory, everything's normal, things are at a steady state, they're fluctuating within that steady state, but all of a sudden, homeostasis is disrupted. A splinter covered in pathogen broke through the skin's protective barrier. The body initially responds to the stimuli through non-specific inflammatory response. The mast cell notices chemicals released from the damaged cells and in turn releases histamine. That histamine travels to the blood vessels, which respond to dilation and they become leaky. Neutrophils, which are bacteria slayers, leave the blood vessel, are drawn to the chemokines released from damaged cells, and engulf and destroy pathogens. The initial immune response, which is specific to the pathogen present, begins when the macrophage here is activated when it engulfs a pathogen and presents the antigen. So it engulfs the pathogen and is presenting an antigen. And all of these antigens on the surface are like normal human um, self antigens. But now it's presenting a foreign antigen. A helper T cell is activated when it contacts the presented antigen on the macrophage. The helper T cell releases cytokines to draw cytotoxic or killer T cells and B lymphocytes to the damaged area. We begin cell-mediated immune response when the killer T cells are checking the presented recycled peptides on the human cells. If the peptides are abnormal, the killer T cells will recognize them as non-self and destroy the infected cell before it can spread the infection. The killer T cell can dissolve holes in the infected cell's membrane, or it can send hormones that trigger the infected cell to go through apoptosis or cell death. In addition to killer T cells progressing through cell-mediated immune response, the human can progress through antibody-mediated immune response using B cells. The B cells are drawn to the damaged area from the cytokines released from the helper T cells and then engage with the pathogen using antibodies, these little Y guys, fixed to their outer cell membrane. This activates the B cell, which presents the foreign antigen. The activated helper T cell comes over, binds to the B cell, which initiates mitosis, nuclear division and cell division. It also initiates mass production of endoplasmic reticulum, and in turn, mass production of protein antibodies. These B cells are now considered plasma cells. The mass-produced antibodies circulate and they end up binding or agglutinating, clumping up the pathogens. They neutralize them so they can't cause infection, and they're marked so that they can be easier to destroy by the eosinophils, which have very similar functions as the neutrophils. Plasma cells and their antibodies are fairly short-lived. So activated B cells can also become long-lived memory cells, which upon second exposure to the pathogen immediately mass produce antibodies. So we've got memory cells that are hanging out that remember this pathogen. So in case we're ever exposed to it again, we can skip all those intermediate steps and go straight to mass production of antibodies. This is the science behind vaccination leading to acquired immunity. All of these processes are essential to maintaining health through responding to infectious diseases from pathogens. We also have scar tissue built up here. That is the gist of how we go from inflammatory responses, which are nonspecific, to cell-mediated immune responses with the killer T cells, and antibody-mediated immune responses using B cells and antibodies. I hope that is helpful.